Antibiotics. The antibiotics unit comprises a large number of antimicrobial agents, which include the following: beta-lactam antibiotics, aminoglycosides, anzomycins, carbapenems, carbapenems, and many more. In this unit, we shall cover in detail the beta-lactam antibiotics. The others will be covered in subsequent units. To start with, it may be worthwhile to ask the question, what is an antibiotic? The noun antibiotic comes from the Greek anti, meaning against, and bios, meaning life. This was suggested by Dr. Selman Waksman, a soil microbiologist, in 1942. Today, an antibiotic is difficult to define satisfactorily, but it may be considered as a chemical compound derived from or produced by a living organism such as a bacteria or fungi, which is capable in small concentrations of inhibiting the life processes of microorganisms. A substance is classified as an antibiotic if it satisfies the following. That is, 1. It is a product of metabolism, even though it may be possible to be synthesized. It is a synthetic product prepared as a structural analog of a naturally occurring antibiotic. It antagonizes the growth and also the survival of one or more species of microorganisms. It is effective in low concentrations. Although many antibiotics have been discovered and isolated, only a few have ever been used clinically. To be successful in therapy, the antibiotic must be A. Significantly effective against the pathogen without serious adverse reactions to the patient. B. That it's stable for storage for long periods of time, which is needed before it is used. C. It is capable of being processed in various dosage forms from which it can be absorbed readily. And D. Not to be rapidly denatured or eliminated from the body. An antibiotic is a medicine that kills or slows the growth of bacteria and is thus used to treat a variety of bacterial infections. Antibiotics are among the most frequently prescribed medicines in modern medical practice. Bacteria are natural, simple one-celled organisms that are found everywhere around us and are needed part of life. Bacteria cause disease and infection only when they are able to get access to more vulnerable parts of the body and multiply rapidly. Antimicrobials, on the other hand, is a larger group which includes antibiotics, antiviral, antifungal, and antiparasitic medicines. The category we are covering now, that is the antibiotics, are divided into two groups. The bactericidal antibiotics, which do kill the microorganisms, and the bacteriostatic antibiotics, which do inhibit the growth of microorganisms. Because there are so many antibiotics, it's been seen logical to put them or classify them, to put them in different classes, which are briefly described now. Antibiotics and antibacterials listed by class. These include aminoglycosides, anzomycins, carbapenems, carbapenems, cephalosporins, which have gone through several generations to the current 5th generation now, glycopeptides, lingosamides, lipopeptides. Also includes macrolides, monobactams, nitrofurans, oxazolidins, penicillins and penicillin combinations, polypeptides, quinolones, sulfonamides, which we have just covered, tetracyclines, and the drugs against mycobacteria, and also a smaller group of antimicrobials which cannot fit in any particular category, and these are measured individually, such as chloramphenicol, phosphomycin, and metronidazole. Examples of each of these are shown against each name of the class they belong to. Before we tackle the antibiotics in detail, just it's important to look at the top antibacterial producing companies by global sales of antibiotics as listed in 2004. The number one ranked company is Pfizer with sales of 2,938 million, followed by GlaxoSmithKline 2,425 million, and the rest in this list as shown here. While we talk about sales of antibacterial, it's also worthwhile to look at new antibacterials that have been developed over the last few years. The introduction of new antibacterials has tapered down to a very small number per year. In 1983 to 1987 period, there were 16 new antibacterials introduced and approved in the US. The following period of 1988 to 1992, that number fell to 14, and by 2003-2005, there were only 3 new antibacterials. Now, the global sales of the major antibacterials as of 2004 are as indicated here, that is led by cephalosporins with 30% market share, followed by quinolones with 19% market share, and macrolides taking 18% of the market share. Penicillins that used to dominate clinical practice by 2004 represented only 16% of the market share. The new class of penems seems to be making impressive gains with 5% market share. Strategy for antibacterial medicines. There are a number of targets on which the different antibiotics act, starting with interference with cell wall synthesis as shown here for a number of these, including penicillins, cephalosporins, glycopeptides, carbapenems, and monobactams. The next category is the folic acid metabolism disruption, which includes sulfonamides and trimethoprim. The other one is interference with DNA winding by DNA gyrase as shown here, done by quinolones. Rifampicins, on the other hand, interfere with the DNA-directed RNA polymerase as shown here. A number of antibiotics interfere with the bacterial subunits, including the 50S and 30S subunits as indicated here, and also oxazoridins affecting the 50S here. Returning to the main topic of discussion for this unit, that is the beta-lactam antibiotics. The beta-lactam antibiotics are of two types, the penicillins and the cephalosporins, as shown here. Both these agents are cell wall active agents that prevent the final step in the synthesis of the bacterial cell wall. In activity, they range from very narrow spectrum to very broad spectrum. Let us take first the penicillins. The first penicillin antibiotic was accidentally discovered by Sir Alexander Fleming in 1929 from a mold, Penicillium notatum, whose plate is reproduced here. As you can see, a colony of Penicillium notatum growing and the bacteria Staph aureus colonies close to the notatum dying off. Since then, a large number of compounds with similar actions have been discovered from different molds. The commercial production of a penicillin antibiotic consists of preparation of a pure culture of the desired organism for use in inoculation of the fermentation medium, fermentation for a given period of time to form the antibiotic, isolation of the antibiotic from the fermentation culture media, then followed by assay for potency, test for sterility, and for absence of pyrogens. Then the final step is formulation in acceptable and stable dosage forms. The diagram here shows schematically that production sequence, that at the top the penicillium and the sugar are added and air is bubbled in to keep the system oxygenated, and if need be, steam or cold water is passed through around the fermentation system to keep an even temperature. When all is done, the culture is removed after the fermentation is complete. The rate of production depends on how much sugar is available. A typical penicillin production plant is shown here. Penicillin spectrum classification. Narrow spectrum penicillins, which are sensitive to beta-lactamase, are indicated here, and those that are beta-lactamase resistant are indicated here and are considered to be the second generation in development. Broad spectrum penicillins, such as which are referred to as amino penicillins, are considered as the third generations and include those listed here. Carboxypenicillins are considered to be the fourth generation, and together with the uridopenicillins. We have summarized the mechanism of action of many of the antibiotics. And we can look at this from the point of view of the site of action, as indicated here, the cell wall, and the name of antibiotic which act on the cell wall, and then the process that is interrupted, and finally the type of activity, whether bactericidal or bacteriostatic. Cell membrane also is indicated as shown here. Those that act on ribosomes, that is the 50S, 30S, are shown here, and those that act on nucleic acids are also, that is DNA or RNA, are shown here. The basic structure of a penicillin molecule is shown here, and where the R equals the unit indicated here, the penicillin is known as benzyl penicillin. The full name of penicillin, such as penicillin G, is indicated here, which is very long. For this reason, an abbreviated form defining a base ring as a penam ring, and therefore the name becomes shorter. One of the oldest penicillins is benzyl penicillin, or Pen-G. The other one is phenoxymethyl penicillin, or Pen-V, as shown here. Following the isolation of those two molecules, a large number of derivatives has been made from them, which include some of those shown here, such as methicillin, oxacillin, cloxacillin, ampicillin, amoxicillin, and carbanicillin. In clinical use, penicillins are rapidly bactericidal, and they are also considered to be very safe. However, there are some problems associated with these molecules. The first one is instability of the beta-lactam ring. The beta-lactam ring is easily degraded by hydrolysis through nucleophilic attack, even by very simple nucleophiles such as water, to give penicilloic acid, shown here. Thus, oral administration of such agents would lead to reduced absorption, as most of them would be destroyed by beta-lactam ring opening. Older penicillins were mainly used to treat gram-positive co-infections. Substitution at the ortho position of the benzyl group, such as methox, an electron withdrawing group, stabilizes the beta-lactam ring. When protonated, aminopenicillin such as ampicillin and amoxicillin also stabilize the beta-lactam ring, the units shown here. The second problem has to do with effects of enzyme degradation of beta-lactam ring. Some bacteria produce beta-lactamase enzyme, which hydrolyze the beta-lactam ring. The bacteria that do so include Staphylococcus aureus. Bulkier groups confer protection of beta-lactam, such as those shown for cloxacillin, dicloxacillin, and oxacillin. Doing so reduces their activity. The other noted advantage conferred by the bulky group is resistance to acid hydrolysis of the product. How do the penicillins work? Penicillins, or the beta-lactam antibiotics, bind to the penicillin binding protein. The penicillin binding protein is unable to cross-link peptidoglycan chains, which are important in cell wall structure. The bacteria then is unable to synthesize a stable cell wall, which then leads to its lysis, as shown in detail next. The bacterial cell wall consists of a complex polymeric material called peptidoglycan, which is made up of both amino acids and amino sugars. The amino sugars are of two kinds: N-acetylglucosamine, NAG, and N-acetylmuramic acid, NAM, which together form a linear polymer of NAG alternating with NAM as shown here. NAG and NAM are linked by a glycosidic bond between the C1 and C4 carbons and are oriented in the same way they are in cellulose. Side chains of 4 to 5 amino acids are attached to each NAM, form covalent bonds with the amino acids on adjacent chains, which then is referred to as cross-linking. So formed covalent bonds may be direct to the next chain in the same plane as shown here, or include additional peptide cross bridges such as 5-glycine residues which extend chains below, as indicated in this stepwise synthesis indicated by this group, which grows to this group, which then links to the unit lower chain to give this particular product. This elaborate covalently cross-linked structure provides the great strength of the cell wall. It also leads to a remarkable conclusion that the bacterial cell wall meets the definition of a single molecule. The beta-lactam antibiotics such as penicillins bind to and inhibit enzymes needed for the synthesis of the peptidoglycan wall. While they have little effect on resting bacteria, they are lethal to dividing bacteria, as the defective walls cannot protect the organism from bursting in hypotonic surroundings, which results in their death. Clavulanic acid. Clavulanic acid has a weakened bacterial effect by itself, but is a potent beta-lactamase enzyme inhibitor and is thus used in combination with susceptible beta-lactam products. Other bacteria produce amidase enzymes which can hydrolyze the amido bond. The NH2 group broadens the spectrum to involve gram-negative bacteria like Escherichia coli, Klebsiella species, Haemophilus species, Salmonella species, Shigella species, and so on. Pharmacokinetics and pharmacodynamics of time dependence. The beta-lactams antibiotics are said to be time-dependent killers. The effect is directly proportional to the amount of time the concentration of the antibiotic at the active site of infection is above the minimum inhibitory concentration of the organism, as shown here. Adverse medicine reactions. Penicillins are considered to be extremely safe and well tolerated by the majority of patients. However, hypersensitivity allergic reactions may occur in 1 to 10%, and these may range from rashes to anaphylaxis at the worst. Penicillins should never be used as topical preparations, as these results in sensitization of the person who uses it in that form. Any contact with the skin by health worker or patient should be washed off immediately. The following extended description of one of the products is given as an example of full product monograph description. This is amoxicillin, which here shows the indications, meaning the types of diseases for which it is considered effective, as listed here. And while using it, also the cautions that should be considered, such as history of allergy, and also includes things like erythematous rashes common in glandular fever, increased risk in erythematous rashes in cytomegalovirus infection, acute or chronic lymphocytic leukemia. In this case, maintain adequate hydration with high doses, particularly during parenteral therapy. Contraindications means not to be used. This would include penicillin hypersensitivity, and also caution to be taken in the case of renal impairment, where crystalluria with high doses may occur. Reduce the dose in severe impairment. Rashes are more common in this case. In pregnancy, is not known to be harmful. Breastfeeding, trace amounts in milk may be seen, but appropriate to use. Side effects include the following: vomiting, diarrhea, rashes. Discontinue in this case. Rarely, antibiotics associated colitis. Also see some is described as a benzyl penicillin. Dose for different patients and diseases is indicated here and continues here. All this has been taken from the BNF 70th edition. Next, we look at the cephalosporins. Cephalosporins were discovered in 1945 by the Italian pharmacologist Giuseppe Brozzo and were first sold in 1964. They are produced by Cephalosporium species. The basic ring is 5-thiaza bicyclo 420 octane, as shown here. As in the case with the penicillins, a base ring has been defined as a cepham, shown here. Together with cefamycin, they constitute a subgroup of beta-lactam antibiotics called cephams. The cephalosporin nucleus, that is 7-aminocephalosporanic acid, was derived from cephalosporin C and proved to be analogous to the penicillin nucleus 6-amino-penicillinic acid, but it was not sufficiently potent for clinical use. Modification of the 7-ACA side chain resulted in the development of useful antibiotic agents, and the first generation cephalotin or cephalothin was launched in 1964. Since then, they have been developed through various generations to the currently in use 5th generation. Some of examples of cephalosporins are shown in this list here. Many of the cephalosporins in use now are semi-synthetic based on the 7-ACA. This change was done to increase the stability in acidic media, to improve pharmacokinetics, especially after oral administration, to broaden the antimicrobial spectrum, to increase activity against resistant microorganisms, decrease allergenicity, and to increase tolerance after parenteral administration. Cephalosporins are indicated for the prophylaxis and the treatment of infections caused by bacteria susceptible to this particular form of antibiotic. First generation cephalosporins are active predominantly against gram-positive bacteria, and the successive generations have increased activity against gram-negative bacteria albeit often with reduced activity against gram-positive organisms. As an example, Cefaclor and its congeners are indicated for infections due to sensitive gram-positive and gram-negative bacteria. The only caution to be considered is sensitivity to beta-lactam antibacterials, contraindications for cephalosporin to cephalosporin hypersensitivity or renal impairment, and not harmful in pregnancy or breastfeeding. Side effects include diarrhea, nausea, vomiting, abdominal discomfort, headache, allergic reactions, and others as shown here, and also additional side effects as indicated here. Reversible interstitial nephritis, hyperactivity, nervousness, sleep disturbance, hallucination, confusion, hypertonia, and dizziness may occur. Some other side effects are also shown here, taken from the BNF 70th edition. All beta-lactam antibiotics act the same way as described before for penicillins. Clavulanic acid. The combination of amoxicillin and clavulanic acid is known as augmentin and is used for a number of diseases as indicated here. That is all for this particular unit.